was actual physiological changes in the brain. So what they did here is they put an electrode in one of those barrels and they wiggled the whisker. These are responses, and you can see that they're dramatically different. The pre-stimulus activity compared here to here is different. The size of the response when the whisker is brought back to rest is quite different, and the response to the whiskers that surround the barrel whose whisker you're deflecting are much bigger here. So now, this is where my lab comes in. We ask, are there underlying structural changes correlated with these changes? Now, of course, giving away the punchline, the answer is yes, because otherwise we'd be done after 5 minutes of my talk. And so what we did is we've been looking at a whole host of things. We first looked at individual neurons. Then we looked at what are called dendritic spines. So if we blow up the dendrites, which are like the antennas for the cells, and look at the individual spines, which are the order of 1 to 2 microns long, see how that is affected by sensory deprivation. We'll then look at 2 different types of glial cells, which are supporting cells within the nervous system. And then finally, we're also going to look at the stuff outside the neuron called the perineuronal net. So this is the paradigm. This is the normal map. This is what we call unilateral trim. Just trim one side, and then bilateral trim. And when we go through this talk, remember, this is what Mickey Mouse has. Notice he has no whiskers. So just remember that when you see what happens, and you think that should he be a role model for our kids. So this is kind of the developmental timeline of the mouse. It gets born here. We begin trimming here. The barrels form by the 4th postnatal day. P stands for postnatal. They open their eyes and they begin whisking around day 10, 11, and then the final trim, and then we sacrifice the animal for the anatomical analysis. So this is a technique called the Golgi technique. It was invented in the 1870s in Europe by Camillo Golgi. Any of you have taken any cell biology classes, you know about the Golgi apparatus. Same. And this is a technique for silver staining. The 2 most amazing things about this technique, first of all, if it worked in 1870, it's going to work at humans, so that's a good thing. The second thing is it was invented in 1870, and here we are in 2017, and we still don't know why it worked. But we're very fortunate, and it fills about 10% of the cells, so you can see here a magnification. So I made the point before that there are kind of 2 main classes of cells, excitatory cells and inhibitory cells. Excitatory cells tend to have triangular cell bodies, and their dendrites are what we call spiny. This is a blow-up of that. As opposed to inhibitory cells, which have smooth dendrites. So excitatory cells mean they cause the cell they're connected to fire. Inhibitory cells cause the cell that they're connected to fire less or not fire at all. So the question is, what happens following trimming in these 2 classes of cells? And I'll go over this analysis because it's kind of interesting. This is now an inhibitory cell, and it looks like it's the middle of a bullseye or a dartboard. And what we do is that this is a 2-dimensional representation, but imagine you have the cell body in the middle and then a series of spheres. Each sphere is 10 microns apart. And what we do is we count the number of times that the dendrites cross these spheres, or we count the length of the dendrite in each of these spheres. The black line represents the control. The white with the dotted line represents the sensory deprived condition. And what you can see, the number of intersections increases following trimming, and the overall length increases following trimming. Another way to do that is to just quantify it in a bar plot, and you can see here the gray is following trimming. The overall length increases, but notice the number of dendrites does not increase. So what this is telling us is that following trimming, the dendrites that they have expand. What happens in the excitatory cell? This is the same story. So there's 2 types of dendrites in pyramidal cells. These are called basilar dendrites, and this prominent one that is going towards the surface of the brain is called the apical dendrite. And what we see with the apical dendrite is that following trimming, the basilar dendrite here is the same exact thing as the interneuron, that they expand. But the apical dendrite is the exact opposite. It contracts. And what's very interesting, when you do a cell-by-cell analysis, the contraction here is equal to the expansion here. It's like the cell has some sort of homeostatic mechanism. So the dendrites of interneurons elaborate following trimming. The apical dendrites retract. You can really see that nicely here. A couple of cells. And overall dendrites within layer 6, which is where we look, expand, while those that go outside layer 6, like these apical dendrites extending up, retract. So we then wanted to focus in, and we're looking now at the dendritic spines. So as I said, this is 2 microns is the scale bar, and what you can do is based on their shape, you can see we identify different types. And so what we then did is in the bilateral trim, we counted the number of spines in the dendrites in control animals and those that were trimmed. And we saw that there was an increase in the number of dendrites. And then we asked the question, the number of spines. We asked the question, if we allow the whiskers to grow back, so we trim the animal for a month, and then we allow the whiskers to grow back for the month, do the animals recover their normal, what we could call phenotype, the normal density of spines? And the answer was no. We then wanted to try to understand kind of what the mechanism of this was. And so we did a pharmacology experiment where instead of trimming now, we're injecting a drug called MK801, which blocks an important type of excitatory neurotransmitter. And what we saw is that when we blocked for a month this excitatory neurotransmitter, we saw an increase in the dendritic spine, just like we saw following trimming. And we think what's happening is when there's less excitatory inputs, that the cell needs some sort of inputs. And so one way in which it responds is it expands its dendrites, trying to find more inputs, and increases the number of spines, where the inputs are being made. So chronic sensory deprivation during development profoundly affects the morphology and density of dendritic spines. And it turns out, I didn't go over it specifically, it depends on what layer of the brain you're in. So the next thing, as I said, we focused on a little bit was on supporting cells. There's 3 main types of glia, or supporting cells in the brain. Astrocytes, which maintain the proper extracellular milieu in terms of ions. We didn't look at those, or we have not yet looked at those. But what we did look at first was something called oligodendrocytes, which produce myelin. Myelin is a fatty substance that coats the axons, the outputs of neurons. It doesn't coat the dendrites, just the axon. And it acts like an insulator, like you would have on an electrical cord. And we use a stain. Every one of these little dots is an axon, is a cross-section of an axon that's been myelinated. And so we asked the question, is following trimming, is there a change in myelin density? Now, you don't need to be a neuroanatomist to say that there's a lot less myelin here than here, and this is where it's quantified. The control is black, and the trim, you can see, is much lower. There are some axons that have myelin, but those axons that have myelin, this is now the diameter of those axons, they have less myelin. And so we asked the question, why is there less myelin? So we have this trimming, we know it's affecting the dendrites of the cells, we know it's affecting some other things. Why is it affecting the oligodendrocytes, which are the number of the cells that produce the myelin? So we do immunocytochemistry, which is a technique in which we can stain for different cell types. And what you're seeing here is blue dots are cells. You can see not every cell then has a black dot on it. The black dots are cells that are stained for a specific protein called OLIG2, which identifies oligodendrocytes. And we counted the number of oligodendrocytes following trimming and see that there was no difference. We then said, well, maybe there's a difference in the number of cells in the barrel, and that would somehow impact the need for axons. And turns out the number of cells are not changing. Now, a laboratory at about the same time we published this paper at Columbia University showed that following trimming, axons have less branches. And if they have less branches, they require less myelin. And we think that's what is happening. And what's the consequence of demyelination or less myelination is if you have less myelin, signals travel slower. All right? And then this is just showing you that what we did is we recorded from a whole animal. We wiggled, instead of wiggling the whiskers, now we use an air puff to deflect the whiskers. And we saw following that air puff in the control hemisphere, which is red, versus the deprived, which is blue, you could see that the response is delayed and the magnitude of the response is decreased, which is similar to other labs findings. So I didn't make this point, but it turns out, and this is true in mice and in humans, is that myelin develops relatively late in life. This is one reason why babies have very poor coordination, because they don't have the myelination. As a result, the neural signals from the brain take longer to get to the peripheral muscles, and thus they're just not as coordinated. Sensory deprivation decreases the amount and the number of myelinated axons observed in the barrel cortex, and decreased physiological responses to whisker deflection. So the next thing we looked at was the perineuronal net, which was first described by Camillo Golgi, and it was what they call the delicate reticular covering surrounding the cell body and proximal dendrite. This is actually a drawing by Golgi's most famous student, Ramon y Cajal, and hopefully this doesn't happen to you guys, but the student wound up hating the advisor, and there was a huge battle. They weren't talking to each other. But they came back together in 1904 because they were the first people to be awarded the Nobel Prize in physiology or medicine, excuse me. And they had to go to Stockholm to receive their prize. So this is what the perineuronal net is. This is a neuron. Here's the dendrites. Here's the axon. The perineuronal net is made of, this is a big word, chondroitin sulfate proteoglycans, which are these blue circles. They have a core protein here, which we're actually going to stain for, called aggrecan. They're connected to the cell through specific receptors, and then there's linker molecules that connect different parts of the perineuronal net. You could see the picture here. They're just kind of ensheathing the cells. The reason why you see nothing in the middle is that within the cell nucleus, there's no perineuronal net. Here's a high magnification taking a confocal, not unlike the one you have upstairs, where the green is stained by Wisteria floribundin, which is actually a protein derived from the wisteria plant, which will, hopefully when spring does arrive, will start blooming around New York City. And then the red dots are the GABA-A receptor, which is the inhibitory neurotransmitter. And what you can see is you have these green circles, and in the middle of them, these red dots. So what we think happens is that the inhibitory inputs get on the cell, they get ensheathed by the perineuronal net, and we think that has a physiological important role, which we're going to investigate in a moment. So, fortunately, one of the first things we did is we wanted to know if the perineuronal net, specifically the one that we had the antibody for, was enriched in the barrel cortex. And luckily for us, the barrel cortex is the highest density of perineuronal nets within the rodent brain. Interestingly, it ensheathes inhibitory interneurons. So the red dots are for an antibody to parvalbumin, which stains a very important, those smooth cells. And it's ensheathed, as you can see, by the green here, which is representing the perineuronal net. And so following 1 month of trimming, if you look at the deprived side, the red dots now are not the inhibitory interneurons. They're actually what's called a Nissl stain, which is a cell stain. It binds genetic material, so it allows us to see the barrels. And you can notice here, ipsilateral trimming means it is the intact side. Notice that you have a lot more green here than here. Now, of course, We can't use that term. We actually quantify that, and you can see following a month of trimming, the overall, which is here, here's the mean, there's a huge decrease in the number of nets, and it does not recover if you let it grow out for a month. A point I haven't made, but we've done this in every condition, but I haven't shown it to you, is that most of the time we're doing this deprivation during early childhood, during the first month of life. So one of the questions, of course, is what happens if you were to do this in the adult? To give you an idea about the lifespan of a mouse, the mouse reaches sexual maturity at about 30 days. So 90 is probably equivalent to like a 30-year-old. P90 is probably like a 25-year-old. Human, they can live up to about 2 years in the laboratory environment. And if we do the same exact experiment in the adult, you notice that we have no effect. Now, it turns out we do have an effect on some of the things, but in general, the brain's ability to respond to changes in the sensory environment are most significant early in development when the network is being set up. Once the network is being set up, changes in sensory activity have effects, but much more subtle effects. So to kind of summarize what we saw right here is that perineuronal nets are enriched within the barocortex. cortex. They're most prominent in the layers that receive the densest inputs from the thalamus, which is a point I didn't go over, and that these co-localize with the parvalbum, which are these fast-spiking inhibitory cells. And sensory deprivation has a specific effect on at least one component of the perineuronal net. So now the question is, can we figure out what this is doing? So we used another technique, which is called path clamping. So what you're seeing here is we can cut a slice of the brain, we can put it under a microscope, we can visualize it now using infrared optics, and you can see here's a neuron, there's a lot of these neurons, and we can bring a pipette down and record from an individual cell in the brain. And this is what happens following a month of trimming. So here, we trim these are now characterized based on their physiological properties. Regular spiking cells are excitatory cells. The fast spiking cells, those are the parvalbum positive inhibitory cells, the smooth cells. And what we see following trimming is that there's very little effect on the regular spiking cells. What we're measuring here is the amount of current we're injecting through the electrode and the rate in which they're generating what are called action potentials, which are how neurons communicate with each other. So there's no change in the RS cells, but the FS cells, the inhibitory cells, you can see following trimming their activity decreases. So we wanted to know, is this change due to the trimming? Well, we know that, but is it due to the loss of the perineuronal network? So one thing we could do is we could trim a lot of animals, and that's very time consuming. Or what my graduate student, Philip Chu, is now doing, did as he graduated, he created a small volume incubator. It's 1 milliliter of fluid, and using an enzyme called chondroitinase ABC in a time-dependent manner, he could digest away the perineuronal net. So the black or very dark brown puncta are the perineuronal net, and you can see after about an hour, the net is almost completely gone. So we're going to digest the net away for an hour, we're going to record from the cells here and also from control slices, and then see what the effect is of removing the net. So there are 5 physiological classes of cells in the barocortex. We're really going to focus on the regular spiking cells, the excitatory cells, and 2 classes of interneurons. The fast spiking cells, which I've introduced, and another kind of cell called the low threshold spiking cells. We're going to record from them in control conditions and then following an hour digestion. What you can see here, this is now something called input resistance. So what we do, recording from a cell, we inject some current and we record the voltage response. The control is in black, the digestion is in red, and what you can see is that the magnitude of the response is red. If you remember from high school physics or Regents physics here in New York State, there's something called Ohm's law. where the voltage equals the current divided by the resistance. It might bring back nightmares for some people. But you could then rearrange that equation is to voltage over current equals resistance, which is what we've done here to measure what we call input resistance. And you can see following digestion, the input resistance decreases. And we think that's because the perineuronal net is kind of acting like a shrink wrap. And if you remove the shrink wrap, you can imagine the membrane is now more leaky. This impacts the firing of the cells. As you can see here, the difference between the control condition and the digested condition, the FS cells show the greatest difference. And that's kind of quantified here, where we look at the red now is the digested condition, the black is the control condition. If we look at the amplitude of the action potential, you can see that there's a large reduction in the amplitude of the action potential. And this effect is very specific to the FS cells, because the LPS cells, the low threshold spiking, which is another class of interneurons, did not show this effect. So it's not a function of the enzyme we're using, it's a function of the net being digested away. So the perineural nets appear to regulate input resistance and spike amplitude of the fast spiking cells. Most active and passive properties of other cell types were not changed by the digestion. And consistent with their cell type distribution, perineural nets impacted the passive and active properties of fast spiking cells more than the other cell types. So how does this impact the network? We wanted to look about the communication between the different cell types. And so we recorded from the cells and we looked at the inputs. And we saw following digestion that the LTS cells, the frequency, if you compare, this is digestion, this is control, you can see the frequency decreases. In fast spiking cells, there's no change in frequency, but what does change is the temporal dynamics. So if you look at the width of the responses in the control condition versus the width of the responses in the digested condition, they're quite different, which we quantify here, where we measure the width of those responses, and you can see that they're significantly decreased following trimming. And there's no effect on the excitatory cells. So what we then did is we could stimulate in one area, the input area of the visual cortex, the thalamus. This black thing is a wire electrode, which we could put in the thalamus. We could stimulate here and then record from a cell in the cortex. And this is very preliminary, but what we are seeing, the chondroitinase is in red, the control is in black. We are seeing some changes. I don't have a lot of confidence. I have confidence we're going to see changes. I don't have confidence in these specific results because they're still preliminary, but I wanted to share them with you. We do think that this is showing us that the net is playing a role in synaptic communication. So the intrinsic properties we went over, but PNNs not only serve as a physical barrier to synaptic reorientation, but also regulate the physiology of inhibitory neurons. And we think this might then impact those results I showed you at the beginning in terms of the physiology. One thing we saw is that the responses went up following trimming. If inhibition goes down, that would be consistent with that. So why are these levels decreasing? One, we looked at it from two aspects. One, we did in situ hybridization with some collaborators at SUNY Upstate Medical Center in Syracuse, and we did an in situ hybridization for the mRNA for aggrecan, which is one of the core proteins of the perineuronal net. And what we saw was that there was a decrease in the message following trimming. That didn't account for all the decrease. We thought there was an active process. So we looked at an enzyme called tissue plasminogen activator, which is a serine protease. Anytime you see ACE, that means it's an enzyme. It's cutting something. It also plays a critical role in degrading neuronal extracellular matrix proteins like the perineuronal net, as well as the modulation of neuroplasticity. This is kind of a funny thing, it says TPA. So professors also sometimes surf the internet, and this turns out was a cartoon character that's used in an advertisement for some sort of potato chip in Japan. I couldn't read that, but I got that in the computer. So we stained for TPA. You can see here, this is following a month of trimming. So everything I've shown you so far is trimming decreases something. Now this is trimming increasing the expression of this protein. It's quantified here where the pink is following trimming, the blue is the control condition. And remember, this is the enzyme that degrades the net. So we think that the net is going down because there's less of the mRNA to create the protein, and there's an enzyme that's degrading it. So sensory deprivation results in increased TPA levels, which are not restored by sensory restoration, perhaps making the cortical environment more permissive to changes, such as the anatomical changes I've shown you. So is the TPA actually getting to the right place at the right time? And we don't know yet, but one possible cell type that could get you there are microglia. Microglia you can think of are the immune cells of the brain. What we saw is here's a control microglia and here's deprived microglia. And you can see two things. First of all, their cell bodies enlarge and their processes retract. These are the anatomical hallmarks of activation in response to an infection or some sort of injury to the brain. And here is the cell body size quantified. Interestingly, if we now allow the whiskers to grow out, they kind of regain the normal phenotype. So what we think is happening is microglia are getting activated some way and then are going to release TPA. And what's the evidence for that? So here's twofold, and this is now suggested. This is, in green, is a perineuronal net cell. In red, we're staining for activated microglia, and you can kind of see the processes interacting with the perineuronal net. And on the right, you're seeing in the red, the microglia, the white TPA, and what we've shown is that following activation, TPA moves towards the membrane of the microglia, and we think it then gets released in the right place because microglia can actually migrate through the neural pill. So during normal sensory development, you have sensory input, a neuron, it releases some TPA, but not enough to prevent the perineuronal net from forming and really crystallizing the circuit as it should be developed. But in the case where sensory input has been deprived, that increases the amount of TPA, which results in less perineuronal net and allows more plasticity. Now, what's getting it there, we don't know, but our working hypothesis is that it's the microglia. So, going back to where we began, you need a lot of time and patience to be a parent, as well as to be a lab head. They grow up, got another daughter who actually presented her science fair project yesterday. But the take-home, if you take home anything from this talk, is that structural reorganization results from sensory deprivation during development and may underlie the physiological and behavioral changes seen in these animals. Now, of course, whenever a lab head stands up here and talks to you about this, they make it seem like we did all the work. Of course, all the work was done by these people. Rade Ramos, who was a postdoc in the lab, who's now at New York Institute of Technology's College of Osteopathic Medicine. Hezong Yang, who was a postdoc, is now teaching in the middle school of New York. Mary Rocco Donovan was a graduate student. Ariana Durando, a graduate student. Rob Steger, a PhD student. Philip Chu, Adesh Vajnet, PhD students. Eric Chen, PhD student. Danny Tam was an undergrad and master's student. Svetlana Abrams, John Kalamovius, Afraz Khan, Alex Pinhas, David Orner were all undergrads, and Areti Siamunis is a professor at Queensborough Community College who collaborates on another project which I didn't talk about. The perineuronal net was started with a friend of mine, Rick Matthews, and his graduate student, Paulette McCrae. And then thanks to all our funding sources. And thank you very much for your attention. So, any questions?
I don't have questions. I'm always alive.
Yeah.
I have a question.
Go right ahead.
First, that was a good presentation.
Oh,
Thank
thank
you very much for sharing
Oh, my pleasure.
I wasn't clear, I'm not sure when you were talking about plasticity. Using the models, you talked about control the unilateral trim and then bilateral trim.
Right.
So my question is, when let's take the unilateral trim for instance, when you trim them